My project was trying to better understand how TIAM2 proteins interact with other proteins and affect cell signaling using something called FRET. Okay, so that's a lot of complicated words, so I'm going to break it down a little bit. So everyone has a bunch of cells in their body, and inside every cell are a bunch of proteins, and each protein has a different job. And so there's one protein that I'm focusing on that has a lot to do with how cells signal each other in the body and what sort of signals they send. So for example, a signal that a cell could send is to tell another cell to divide or to tell a cell to move somewhere in the body. So the protein I'm focusing on has a lot to do with cancer and the metastasis of what causes it. So when a protein inside a body gets a signal, it can take that signal and change it and send it elsewhere in the cell, and it can kind of manipulate that message. So if something's wrong with that protein, it can manipulate that message and turn it into something bad, which would cause the cells to multiply too much, which is what would cause cancer. So the idea of this project is that if we can kind of better understand how this protein works, then maybe we can do something to stop it from sending out that signal that causes this cell metastasis. So what better way to understand the protein than to create a version of it myself that I can run tests on? So what I did is that I decided to make my own protein, and every protein is made of DNA. So DNA is the stuff in your body that kind of makes up who you are. It determines if you have blue eyes or blonde hair or if you can roll your tongue. So the protein that I'm interested in is made of 3 main pieces of DNA, and my job was to get these pieces and put them together to form this one protein. So I'm trying to make this protein made out of these 3 components here. And this sounds simple, but it has quite a few steps to it. So the first, I had to get all of the pieces of DNA that I needed. And these DNA pieces were housed in much longer strands of DNA. So I had these 2 strands of DNA, and I was only after this piece, and this piece. And then I was going to create those 3 pieces to make this one. So to get the protein pieces I want, I had to use something that acted a little bit like DNA scissors. It found the pieces of DNA that I wanted when the bigger strands, within the bigger strands of DNA, and it made little cuts so that I could cut out the pieces that I wanted. Next, I wanted to be sure that I actually cut out the right pieces. So I ran the pieces of DNA through a gel, and depending on how big the pieces of DNA were, that's how far they would move through the gel. So you can see here that I had the gel, and you can see little bands in the gel. And if the band was a little smaller, then it would move a different distance than if the band was a little bigger. And since I know how big the pieces are that I wanted, I could tell, depending how far the DNA moved, if it was the right piece that I was actually looking for. So after making sure that I had the pieces of DNA that I wanted to put these little DNA pieces together. However, these DNA pieces are kind of like puzzle pieces. If the ends don't match up, then they're not going to glue together to form this concentric circle of DNA. So it'd be like if you tried to fit like a triangular puzzle piece with a circular puzzle piece. Like it won't go together right. So I had to do a special procedure called PCR that makes all of the ends kind of match up so that I can glue them in this big circle. Now before I made this circle of DNA, I wanted to do one more test to make sure I had the right pieces of DNA that I wanted. So the test was called a transformation test. So you know how when you're sick and you go to the doctor and they give you an antibiotic, and the antibiotic will kill whatever germ you have? This was kind of a similar type of thing. I mixed the DNA with some cells, and I put these cells with the DNA on plates containing antibiotic, and depending what piece of DNA I used for this process, the cells either should have grown on the antibiotic plates or they shouldn't have grown. Now you can see here that there was growth on every single plate, which wasn't a good thing because some of the DNA I used wasn't supposed to be able to grow in the presence of the antibiotic. So I was kind of in the middle of troubleshooting why this was happening when I ran out of time for my project. The end goal of the project, what I would have loved to have reached, was to do this process called FRET that I mentioned earlier. And FRET stands for Fluorescence Resonance Energy Transfer. It's kind of a lot of words, you just have to remember FRET. And how that works is you take a protein, and a piece of the DNA that I was after to create this protein lights up a color when it gets excited by energy during fret. So the light goes in and it lights up this protein, and then when the protein is interacting with another protein in the body, so like when the cells are interacting, kind of sending a message saying, hey, you should do this, you should do this, then that other protein will light up too. And so scientists can use those two lights and some other factors and kind of understand how the two proteins are interacting. So that's a little summary of my project. I hope you enjoyed it and learned a little bit today. Thank you.